Thanks so much. I am so delighted to be here with you this morning. I'd like to thank Dr. Lala as well as Dr. Davies and the program committee for the kind invitation to join you today to talk about a topic that I care about and have been working on for some years now, which is the use of patient-reported outcomes for monitoring toxicities and symptoms related to disease in oncology. And I think it's fitting that there's also a CAR T session this morning because part of the U.S. Medicare coverage determination decision for allowing CAR T therapies to be used in the U.S. will actually likely require collection of patient-reported outcomes over time to understand the effectiveness as well as the toxicities of those therapies in the real-world setting. So it's a nice pairing. These are my disclosures. So our session this morning really speaks to the topic of the evolution of therapies in oncology. And as we think about many of our careers, how they've spanned from the continuum, starting with cytotoxic chemotherapies to the kinase inhibitors, and now more recently to immunotherapies and other biologics, the way that we think about toxicities and the dynamics of toxicities has fundamentally changed. And therefore, the way that we think about population management and the way that we think about symptom assessment and treatment has invariably changed and needs to change. And to my eye, this really underlines the importance of monitoring and managing symptoms. And not only has this been of durable importance, I would argue that it is of increasing importance because with some of the newer agents that people either take at home when they're away from the clinic or that lead to long-standing, low-grade toxicities that may impact the tolerability of treatments, adherence with treatments, the patient experience, and ultimately outcomes is ever important. So for me, I often go back to this kind of information. So I'm an oncologist, and so when I sit down with patients, one of the first questions that patients will ask me is, how is this therapy we're talking about going to make me feel and function? And one of the key data sources for this information is what you see on this screen. This is a toxicity or adverse reaction table taken directly from a US FDA drug label. In this case, it's for docetaxel, which is a cytotoxic, but the same kind of table applies to all of the classes of therapies that we may see, including the newer therapies. And what's interesting to me about this is, if you look at it in the first column, you see what we call adverse reactions. These are all of the adverse events or toxicities that the developer of the product and the regulatory agency together have decided are most important. They're either common or they're impactful on patients. In the next column, we see the proportion of patients throughout the course of the pivotal trial that led to the registration or the approval of this drug who experienced any level of that toxicity. And then we see in the last column grade 3 and 4 toxicities. These are the severe or the very impactful magnitudes of each of these. But to me, what's striking when you look at this kind of table is how many of these items we might classify as being symptoms or symptomatic toxicities. Many of them are highly subjective phenomena that we all know in this room are best known to the person who is experiencing these toxicities, the patient, right? If you look at the list, you see items like nausea, taste disturbance, dyspnea, fatigue, myalgia, right? These are highly subjective. But when you think about the processes by which information about these experiences that patients have are collected and documented, it's not by the patients themselves, it's by us, or most of us, who are professionals. It's either by clinical investigators or by our administrative staff members in the case of clinical trials, and it may be by other individuals in clinic visits as well. And I'm going to start by talking about processes in clinical trials. I'm going to end up by talking about processes in routine clinical care. So in oncology, what is the standard current approach to toxicity monitoring? It's a tool called the CTCAE, or the Common Terminology Criteria for Adverse Events. Some of you may be familiar with this. This is a lexicon or an item library that's designed for professionals to complete. It consists of about 800 individual items, And about 10% of these are symptoms, and I'll come back to that a little bit later. And they're all graded on the kind of scale that you see on the screen here. So each individual item, which can be used one at a time, it's not used all together, can be selected for grading. So here you see the CTCA item for stomatitis or oral mucositis. You see that there are 4 grade levels. 0 is implied. And you can see that for each of these, they are anchored to discrete criteria. So if you look, for example, at grade 2, stomatitis, you'll see this is defined as moderate pain not interfering with oral intake, modified diet indicated. So for any of you who are measure developers, right, this is like a classic multi-barreled item, right? So you have severity, you have interference, you have sort of clinical judgment in there, but nonetheless, this is the law of the land. This is how it's done. So early on, as my interest in this area developed, I became curious about whether this CTCA tool is reliable for evaluating symptomatic toxicity, since this information is used to evaluate the risks or the safety of drug products. So is it actually a, is it a clinical trial grade reliability? And my postdoc at the time, Thomas Atkinson, who's now faculty at Memorial Sloan Kettering Cancer Center, did a simple experiment where he had 2 different oncology clinical investigators evaluate the same patients at the same visit for the same drug without being privy to each other's evaluations, and both of these investigators or clinicians used the CTCA criteria to evaluate these symptomatic toxicities. And for clinical trial grade data, we would want to see high reliability of metrics. We'd want to see interclass correlation coefficients in, I'd say, 0.7, 0.8, even higher range. But unfortunately, as you can see, the ICCs across the board for these are lower than we would like to see, right, in the anywhere from sort of, the high 0.4s to 0.5s or 0.6s, suggesting that if, I and one of my colleagues see the same patient at the same time, there's going to be some noise introduced into this metric, which makes one scratch one's head a little bit. Now, what is of further concern, though, is really the process by which this information is gathered and reported in clinical trials or in drug development. And it's worth taking a moment to think about what that process is. We think about drug development. Drugs these days are often developed in many different countries, in many different languages, with many different processes that need to be all standardized. And so what happens is a patient who enrolls in a trial comes in and they're experiencing a symptom or a side effect of the drug. And a clinician, like one of us, interviews the patient, and then they will document that into the chart. So we now have sort of a clinician interpretation and now a chart representation of that symptom, assuming it even makes its way into the chart. And then a data manager needs to come and abstract the chart. Some of these are electronic, some of these are on paper. Often the terminology used in the medical chart is not exact to the CTCAE or to MEDRA, which is another common tool that's used for evaluating adverse events. And then there needs to be manual data entry into a database. Now, in this game of telephone, there are many opportunities for data loss and data transformation, and I would argue that direct patient reporting simply bypasses this entire process and provides for us unaltered the patient experience with disease and treatment. Yet this has not been the standard approach, at least not in the past. But there's a whole host of technologies available to us now for collecting patient-reported outcomes, both for understanding symptoms related to disease and understanding toxicities related to treatment, including these modes that I show on the screen. So again, early on, when I became interested in this area, as many of you have been interested in this area, I think you probably all have a shared experience that I've had, that there are many skeptics out there about some of the things that we do. And one of the questions that the skeptics posed was about the agreement or the comparison of patient reporting of this information and clinician reporting of this information. And what was stated to me is, look, we're clinicians. We understand our patients. We know what's going on with them. We have an excellent idea of what their symptoms and their adverse events are, right? That's what we do, right? That's our job. And if, in fact, that was the case, that we as clinicians did an excellent job of understanding and recording our patients' toxicities, then that would mean that patient-reported outcomes are really unnecessary because what we already do in our current processes is adequate. So my group and other groups embarked in a series of experiments comparing patient and clinician reporting. And I'll show you results from one such of those studies. This was a longitudinal study in which patients receiving chemotherapy and their providers both reported the analogous CTCAE symptomatic toxicities over time, in this case for more than 2 years. And these curves show the cumulative incidence of these various symptoms over time. And you can see the patient-reported cumulative incidence in orange and the clinician in blue. And you can see that across the board for these common and important symptoms, there's a substantial delta between the curves. That space represents what we are missing that our patients are experiencing, at least according to the patients, if that's considered the gold standard. I consider this to be a lost opportunity to understand what our patients are experiencing. But I think of greater concern is this is the information that is missing when a regulatory authority at the FDA or the EMA or wherever sits down to balance the risk and the benefit of the products that our patients are going to be exposed to in the market, because they will have an underestimation of the true risk associated with the product because we're not capturing that information. Now, the next question that the skeptics came to me with was the feasibility of doing this, right? People would say, well, Dr. Basch, this is all well and good. Sure, you know, the patients catch more than we catch, and, you know, maybe we're not totally reliable. But, you know, we're there, and we're able to report this information. In fact, we do report this information. There's a reason organically that the way that we do things has evolved, which is that our patients are sick, our patients are busy, our patients have a lot going on. They're really not in a position to be reporting this information. They're probably not willing or able to do it. So, you know, this then meant a whole series of feasibility assessments to show that patients are, in fact, willing and able to do this. And so there have been many studies at this point in this area demonstrating feasibility, but I'll show you data from one such study, which was an embedded correlative trial to 9 large multi-center US and Canadian based cancer clinical trials showing very high rates of patient compliance with self-reporting during enrollment in clinical trials. This is somewhere in the mid-90% range. And there was actually a recent report, many years subsequent to this study being done, showing that many of the trials submitted to the FDA and the EMA actually have similarly high compliance rates. So you know, patients are actually enthusiastic to self-report. And in the debriefing work that we and others have done, it turns out that patients feel more engaged with their care or with the trial. They feel more connected to processes of care when they're being asked about how they're feeling. So one approach that some have been interested in is sharing patient-reported outcomes in real time with clinicians. The argument being, well, you know, maybe the problem is that there are all kinds of inherent aspects of the interpersonal dynamics of care that prevent us from really understanding how patients are doing. And maybe if we used electronic data collection approaches to put that information in front of clinicians, maybe the clinicians would become more aware of this and there would be more concordance between reporting. And so I'll show you results from a study that was done across multiple phase 2 clinical trials where patients were split into 2 groups. So all the patients self-reported using electronic PROs. For half of the patients, that information was not shared with the clinical investigators, and for the other half, that information was shared with the investigators with this interface. And I apologize to those in the back, it's a little small, but I'll explain. Patients would self-report their own toxicities. Then this interface would be shown to the clinical investigators. On each line is an individual symptomatic adverse event, so you know we have cough, dyspnea, epistaxis, etc. These are the grades reported by the patients. The clinician could either agree or disagree with the drop-down, and if they disagreed, they could reassign the grade. And then we simply looked at the agreement between the patients and the clinicians on the grading of these toxicities. First, we looked at agreement between the patients who reported and the investigators who did not see what the patients reported. And what did we see? Lo and behold, the same exact finding as in those curves that showed discordance. Right? The clinicians are missing most of what the patients are experiencing. Here we see in the middle with this blue shaded part of the bar, the proportion of patient-clinician pairs that actually agreed on the CTCAE grading for these important symptoms, 55%, 50%, 57%, 22% for performance status, right? Very low agreement. And in cases of disagreement, the patients tend to grade higher than the clinicians. But then when we compared the concordance between patients and clinicians for those pairs where the information was shared with the clinicians, we saw extremely high concordance. Again, the blue bars showing the proportion of patient-clinician pairs for each of these individual symptoms for which there was absolute agreement in the CTCAE grading between the patients and the clinicians, on average about 94% agreement. So we as clinicians or investigators, when faced with the self-reports of our patients, will tend to agree with or endorse what our patients report. And in debriefing of the clinical investigators involved with these studies, it was found that the investigators felt that indeed they thought that the patient report was the gold standard. It provided clinical trial grade data. They believed what the patient said, and they're actually grateful to have that information to aid in their own grading. So based on this work, the National Cancer Institute became quite interested in modifying the CTCAE to include the patient voice, and starting in about 2008, the NCI issued a series of contracts to develop a PRO version called the Pro CTCAE that Dr. Lala alluded to in his introduction. And actually, the most recent of these contracts is still ongoing now and is more involved in data visualization and data interpretation. I had the pleasure of my group leading this work, but it was really with a very large and diverse multi-institutional group of collaborators that this work was done. And the Pro-CTCAE is the intellectual property of the U.S. National Cancer Institute. This is a public property that can be used by anybody in this room anytime, and I'll show you the website for that in a moment. So the Pro-CTCAE consists of 78 adverse events. Again, this is about 10% of the CTCAE, right? A PRO for a toxicity like retinal detachment doesn't really make sense. I mean, not completely, but it does certainly for, say, nausea. Each of these items has a generic item structure, and then for each individual adverse event, there can be up to 3 individual items representing the attributes of frequency, severity, and interference with daily activities. And so this is what the conversion from CTCAE to Pro-CTCAE looks like. You start out with a CTCAE item. This is exactly what I showed you earlier, and you wind up with this for mucositis, 2 individual items. The first asking, what was the severity of your mouth or throat sores at their worst, and how much did mouth or throat sores interfere with your usual activities? We break it down into individual domains. We don't multi-barrel this, and we have very simple verbal response options for each of them. When there are multiple individual items, there's a composite scoring algorithm that can bring these all back to a single Pro-CTCAE numerical score. The Pro-CTCAE has been extensively cognitively tested, both initially in its development and then subsequent to its development, to assure patient understanding at many levels of education and health literacy across languages and cultural orientations, and to make sure that the terminology maps to the concepts of interest. In addition, we did a very large validation or quantitative testing study. This was based in the US, although now there have been validation studies ex-US as well, in which each individual item was individually tested. We didn't test it as a piece. So we looked at each item for its construct validity, reliability, responsiveness, the appropriate recall period, et cetera. So this means that you can go into this library and pick individual items. You don't have to use it all, as one tool. You can use which items are appropriate to a given setting. I'll give a quick example. The Pro-CTCAE now has been used in many, many trials in the industry, in academia, in the cooperative groups. This is one example from one of the early trials. This was a study done by the sponsor Exelixis, testing their drug cabozantinib compared to mitoxantrone in metastatic castration-resistant prostate cancer. The investigators chose 10 individual Pro-CTCAE adverse events. These were administered every 3 weeks to patients at home between visits using an automated IVR telephone system. And if patients did not self-report, a human would call them to administer the items, although that was hardly ever necessary. And average compliance, again, very, very high. It was 96% overall at each time point, which is really telling because this was a very sick, heavily pretreated population. Many of these patients were close to hospice or death, and yet the rates of compliance were very high. So I'm going to show you from this trial the between-arm comparisons. First, this is exactly what I showed you in that very first table from docetaxel. This time it's for this trial. Oops, sorry, I want to go back. So this is a toxicity table using the CTCAE, right? This is investigator reported. This is not the patient. This is the standard approach. This is for grade 3 or 4 for these individual symptomatic adverse events. What we see is the proportion of patients in the cabozantinib arm and the mitoxantrone arm for whom their investigators reported grade 3 or 4 toxicities for each of these. You can see the rates are pretty low, a little higher for fatigue, and none of the differences between arms are statistically significant. Here is the pro-CTCAE. This is the patient self-reporting. So first of all, we see that the rates are higher, substantially higher. But to me, what's striking is the difference between the arms. Look at diarrhea, right? 44% versus 11%. And for 4 of these adverse events, the differences between arms is highly statistically significant, suggesting that the precision of the patient-reported tool for understanding the delineation between arms in terms of toxicities is greater than the clinician report. It's a better tool. The Pro-CTCAE is available publicly. You can use it now. You can download it off of this website, which is available from the U.S. National Cancer Institute in multiple languages, and there's also background information available. For the last few minutes, I'm going to talk about the use of patient-reported outcomes in routine care. Many of us in this room are involved in symptom management and assessment in the routine care setting, and Dr. Davies had asked me to touch upon this in my talk. Now, why is this important? I think it goes without saying to this audience that toxicities and symptoms of disease are common in oncology clinical practice, and there is already prior research that shows that patient-reported outcome monitoring during cancer treatment can improve communication, symptom management, and quality of life. Again, we have these electronic tools for collecting information that we might also use in a clinical trial, but in clinical practice, we can now deliver longitudinal symptom reports to providers at the point of care, showing them trajectories. And we can also send real-time alerts to providers about symptoms that are worsening or are of severe magnitude. For example, this one's shown on the screen. These can be triggered to email, they can be sent by text, or these can be interfaced into electronic health record as part of a shared-in basket. Moreover, in clinical care, you can link the patient-reported outcomes to symptom pathways. So when a patient self-reports a severe toxicity, they can get a real-time message back to them when they're at home about how they can manage their constipation or their diarrhea. If this is interfaced to the electronic health record, you can even have some intelligence in the system, knowing that they have a PRN for their nausea or their constipation or whatever. Moreover, when, say, a nurse or a care coordinator or a physician gets one of these alerts about their patient, they can also get a symptom pathway, improving, right, the appropriateness of the delivery of information for management of symptoms. I'm going to describe in my last couple of minutes a trial that my group reported at ASCO and in JAMA 2 years ago. This was a randomized controlled trial called the STAR trial, in which patients who had solid tumor malignancies were randomly assigned either to self-report their own toxicities using the web, with automated alerts sent to nurses for severe worsening symptoms, and these printed reports provided to their nurses and oncologists at visits, compared to a control arm, which is a standard way that we do things. And patients were followed throughout their entire treatment course, and we measured a host of outcomes. This trial involved 766 patients who were enrolled through 2011. They were followed until final analysis in 2016 with a median follow-up of 7 years, at which point, sadly, 2/3 of these patients had passed away. Compliance with treatment, or again, feasibility, was high. Not as high as in clinical trials. This is all comers. This is routine care. But on average, about 75% of patients will self-report their own symptoms during any given time during routine care for long periods of time. So this is the result of our quality of life assessment. We assessed this using the Euroqual EQ5D for a host of reasons. And what we found was that assessed at 6 months compared to baseline, compared to standard care, 31% more patients in the self-reporting arm experienced quality of life benefits. We saw this both in terms of the proportion of patients who had improved quality of life at 6 months and a decrease in those who had worsening quality of life at 6 months. We also looked at utilization of services, hospitalization, ER visits, et cetera. This is the result for the ER visits. And compared to standard care, 7% fewer patients in the self-reporting arm visited the ER with durable effects throughout the study. And perhaps most striking is the overall survival analysis, showing that compared to standard care, median survival was 5.2 months longer among patients in the self-reporting arm. And this translated into a 5-year absolute survival benefit of 8%, which the discussant on this presentation at ASCO noted was a greater survival benefit than all but one of the oncology drugs that had been approved by the FDA in the prior year, but with less toxicity. So several mechanisms of action are attributable to this effect. One is that proactive monitoring prompts clinicians to intervene early before symptoms worsen and cause serious downstream complications. Symptom control enables patients to stay more functional, which is known to be associated with better survival. And symptom monitoring enables control of chemotherapy side effects. And in fact, we found that on this trial, patients who are self-reporting their own symptoms were able to stay on chemotherapy on average 2 months longer than patients in the control arm. This finding has been duplicated in this study, also published in JAMA earlier this year by my colleague Fabrice Denis in France. This was a randomized controlled trial in patients with lung cancer showing an even greater overall survival benefit in an arm of patients self-reporting their own symptoms. This is ongoing work that we're doing. This is a national US trial that's open at 52 sites currently, where we are replicating these results and also looking at strategies for implementing PROs into routine clinical oncology care. So in conclusion, patient self-reporting improves monitoring of toxicities and symptoms and outcomes in trials and routine care. It expands our understanding of the patient experience and engages patients in research and care. I would argue that this model applies not only to our early cytotoxics, but to the drugs that are being developed today. But the work ahead, I feel, is in implementation, integration with electronic health record systems and clinical pathways, and coordination with palliative care, navigation, and care coordination programs. I'd like to thank you very much for your time and attention.